Okay, today, as promised, we're going to talk about different forms of dominance. Before, the only example we used was complete dominance, as in if there's even one dominant allele, or in our Punnett square example is a capital letter, that is the version of the trait or the allele that phenotype masks or covers up or dominates the recessive allele. So say you get a big T from mom and a little t from dad, even though you have that recessive allele, it's not expressed. The big T you got from mom is expressed, and maybe you have a creep toe, or you can roll your tongue, or whatever we use, the T to stand for in different examples. So those were called Mendelian examples because a lot of what Gregor Mendel saw in the pea plants were forms of complete dominance. Today, the different forms a lot of times are referred to as complex patterns of heredity or non-Mendelian genetics. So again, up till now, we've only discussed one version, which was complete dominance, but as to be expected, more complex patterns of heredity can include polygenic inheritance, incomplete dominance, multiple alleles, and codominance. So there's one example, not necessarily an example from a non-Mendelian trait, so it may not fit here, but I use this as an example to help you guys with your word usage. The phrase poly means many. Genic or genetic would be many genes controlled at one trait. And this is an example of polydactyl, which we talked about before, big P being a dominant trait or a dominant phenotype. You can see how there's many fingers or many toes. So again, just trying to give you guys examples. If you ever don't understand a word, try to break it down and look and see what those different parts mean. Another clear-cut example of that was when we talked about photosynthesis last semester. Photo, light, synthesis to make, so you put that together, it's making things with light. So just another reminder there. The first was polygenetic inheritance, and this is going to be any time where there's multiple versions. So instead of tall versus short in pea plants, or purple versus white flowers, or yellow versus green seeds, or round versus wrinkled seeds. Here, anywhere where you've got multiple variations, or instead of and or, it's everything in between, things like height, weight, skin color, hair color, and the example given here is eye color. So it's a trait caused by several genes. These genes can all be on the same chromosome, or they could be spread out over a bunch of chromosomes. The point is that once expressed, they work together to control the phenotype. The phenotype example here is eye color. You can see that here's blue eyes, like what I have. Here's kind of in the middle, some people will call that hazel. And then here's brown eyes. So if you look at the diagram, these are melanocytes or color cells within the iris, which is the ring around the pupil, between the pupil and the sclera. And the more melanosomes you have, the more pigmentation you have in your eyes. So you can see how brown eyes have a lot more melanosomes versus blue eyes. So examples, again, height, weight, hair color, skin color, eye color. Again, anything where there's just a myriad or plethora, I suppose, of phenotypes, variations in between. Instead of yes or no, there's version A, version B, version C, version C, version D, version E, everything in between. So this isn't very scientific again, but just to show you some of the different outcomes. With polygenic traits, you can't always predict really easily what's going to happen. Your Punnett square would be a dihybrid or trihybrid or even beyond that cross. It's not as simple as, well, if dad has brown eyes and mom has blue eyes and the baby has brown eyes. All right, brown isn't necessarily as dominant as one would think because it's not, coded for by a single gene. Again, it's polygenetic. So, for example, I have blue eyes, my wife has brown eyes, and this chart says it's a 50-50. It's probably close to that, but not exact. Our daughter together has blue eyes. So, a few examples there. Next would be incomplete dominance, and once again, break the name down. As opposed to complete dominance, like we talked about before, where the presence of a dominant allele completely covers or masks or dominates that recessive trait, incomplete dominance means neither version or neither allele is completely dominant over the other. So they both try to express themselves. So example here would be anything that results in an intermediate or middle phenotype. Easy way to think about that would be instead of red or white, you would have a blended effect, an intermediate phenotype, which would be pink. Instead of brown or white, you would have tan. Instead of black or white, you would have gray. Instead of long or short, you would have medium. That's what that intermediate is referring to. So again, an example here, I've told you guys before that I used to work in a flower shop before I was a teacher, one of my mini summer jobs, and we sold flowers known as 4 o'clocks. Well, they could be red, they could be white, or they could be pink. And what was interesting about these flowers, these aren't necessarily 4 o'clocks, they're just examples. I think one of them's a New Guinean patient, and the other one's, doesn't matter. Okay, so these are examples here. And you could always tell by looking at it what that genotype was going to be. So unlike what we told you last time, here sometimes you can judge a phenotype by the genotype or judge a genotype by the phenotype. You know, a lot of times we told you not to do that. But here, you know, in an incomplete dominant situation, it could only be certain things. Red would be, for example, big R, big R. White would be little r, little r. All right, and then pink would be one of each, or heterozygous. So one would be homozygous for one allele, one would be homozygous for the other, and then the intermediate would be the heterozygous form. Other examples of flowers would be snapdragons, can be yellow, red, or orange. Grasshoppers can have long, short, or medium legs, and in hair can be straight, curly, or wavy. Here's an example again. So here we're using big R's and little r's. Again, because it's an incomplete dominant situation, the little r isn't necessarily recessive. It's just using that lowercase letter to show another version of the allele. You could just as easily say, you know, big R for red and W for white, and then RW as a heterozygote would be the intermediate form. So you could do it either way, and I've seen it written and expressed either way as well. So if you look at this cross between two heterozygotes or two pink flowers, the 1/4 of them, this box up here would be big R, or one version and one version. So homozygous, so it would be red. Just like down here, it's homozygous, but it's white. It's the two other versions of the allele. And then you'd have 50% or 1/2 of this phenotypic ratio, which would be one of each, or the heterozygote. Codominance is similar to incomplete dominance, where you could tell what's going on as far as the genotype and phenotype are concerned. But unlike incomplete dominance, where neither allele can completely cover the other up, here they're both equally expressed. They're both co-dominant. So you can think of a cooperative or like co-captains on a softball team. So when two dominant alleles are displayed at the same time, we have a co-dominant situation. So here is an example where you would, you could think of like cows, or if you guys ever been in the country and seen, spotted cows or horses or sometimes dogs. It depends on the breed, things like that. So there's roan cows, which would be, reddish brown. So you've got a version that are all white. You've got a version that are all dark brownish red, but if they're the heterozygote, they would be what's known as roan, which is white with splotches of red, or maybe it's red with splotches of white, who knows. Another example, again, I've told you guys a lot of times that I've grown up on a dairy farm. Well, milk cows are known as Holstein cows or Holstein cows, and they look like this. That's your traditional type of cow you see in a cheese commercial or something like that. But there's actually black versions of these. There's white versions, but the heterozygous, if they get a different allele from each parent, if there's a white Holstein and a black Holstein cow having offspring, well, they're all going to have one version of each allele. 100% of them are going to be the heterozygous genotype, which means they're going to have this codominant or heterozygous phenotype as well, which is white with black splotches. You can see some in the background here as well. So this would be a milk or a dairy cow. Finally, there's multiple alleles. I told you before that we use letters a lot of times with this because there's two discernible forms, or easily distinguished between big and little letter, big A, big T, big F, little f. Well, that's because a lot of times genes are the same way. There's, a dominant versus recessive, or maybe even if it's codominant or incomplete dominant, there's two forms. Well, there's not big A, little a, and medium A. There's not always a way to easily represent the third allele to the party. So multiple allelic traits are traits controlled by genes with 3 or more alleles. So not very many examples of this. One would be blood type. There's type A, there's type B, and there's type O. Any guesses as to what AB would be? Well, A isn't completely dominant, neither is B. So, but instead of blending or mixing, they're actually codominant. So A, B, there's type AB, which would be the heterozygote, and then there's type O, which O is really more of a 0. It means you don't have either A or B proteins in your blood. And we'll show you an illustration here in a moment. So here's the ABO blood system. If you're type A, you've got A surface proteins. Type B is B surface proteins. As I just said, O would be none. And then type AB, again, they're codominant, so they're both. Right? So Here's another example. We use these little things, not necessarily, sometimes they're called carriers, but it's just a way to carry the letter. We use I's. So type O would be I's that you get from each parent without A or B. So your phenotype then is type O. You've got type A. There's two ways to be A. You can be homozygous for A, which is you get an A from mom and an A from dad, or you can be heterozygous from A, which means you get an A from one parent, but the other parent was type O. So you get here and here. You get type B the same way. You can be homozygous for B or heterozygous for B. Type AB, you have to get an A from one parent and a B from the other. So because you can't have three versions of the letter B or three versions of the letter A, we use these little things called carrier letters to carry each of these genotype just denotations, I suppose. So then you get into blood donation, and we'll do a blood typing lab in here. No, we're not going to use our real blood. You never know what could happen there. But we'll use some fake blood and we'll type it. So a lot of questions about blood type always. The most common is O positive. Disclaimer, that's what I have, O positive blood. The other most common is A positive, and you can see the rest of these are awfully rare. And it's also broken down. There's different versions or different phenotypes of blood are more or less prevalent in different ethnicities. For example, Hispanic, over half of Hispanics out there have O positive. Here you've got less than half of both Caucasians and African Americans are O positive. So just one example. A lot of people will ask about, blood donation versus blood reception. Well, type O would be that universal donor because there's not any antigens or anything like that on the blood cell itself, this erythrocyte. You could actually give to anybody because none of these are going to pick up on it. Whereas if you're type A, you can give to A. If you're type B, you can give to B. If you're type AB, you can't really give to A or B, but you could receive from anybody. So again, there's more intricacies involved there, and we'll talk about that later. The last question about blood type is always, where does that positive or negative come from? And that's for Rh factor. Positive means you have the Rh factor with your blood. It's just a type of protein. I think it's Rh because they first discovered it in rhesus monkeys. Negative would be you don't have that. That's another thing that's passed on. It's separate. So in some instances, you could think of this as being polygenic because there's a lot of different genes that go into coding for your blood type as well. But, example there, I have type O positive blood, as does my wife. Our daughter is O negative. That means we both had to have been heterozygous for O positive. If we both passed on that recessive allele, there was a 25% chance for my daughter to be O negative, and that ended up being the case. So type A, there's that antigen A on the red blood cells. You've got B antibody in the plasma. Type B is the opposite. AB, you've got both antigens on your blood cells, so you've got neither antibody in your plasma. That means nothing's going to attack its own blood. Type O, you've got both those in your plasma because you have neither on your red blood cells, which means type O can only receive from other type O because they have these antibodies in their plasma that would attack A, B, or AB if given.